starting with the name of Almighty Allah, the gracious and most merciful. Today now we will starting the chapter number 1 and lecture number 5. Topic is tissue culture. Tissue culture. Tissue culture. Tissue mean is potent. Culture mean agar. First one is genotypes. The second one is phenotype. Definition tissue culture. The process or technique which is used to complete tissue from a single cell called tissue culture. Tools required for tissue culture. Tools required for tissue culture. The first one is Lemes flow. The second one is incubator. The third one is water bath. The fourth one is luminous intensity. The fifth one is centrifuge. The sixth one is sterilizer. Temperature. The cell or tissue to be cultured has some tissue culture points. The cell or tissue to be cultured, the cell or tissue to be cultured has some genotypes. Genotypes of mother cell. Some genotypes of mother cell. Second one is the meristematic tissue. The meristematic tissue is puzzle mostly used in plant. Plant cell are totipotent. Plant cell are totipotent. Plant cell are totipotent. its main points of tissue culture. Now we discuss the tissue culture diagram. Tissue culture. Tissue culture. Culture, structure. First of all, we want to ideal leaf. It's a leaf. Okay. Ideal leaf. Ideal leaf. It's ideal leaf left to the water bath. It's a water bath. Remove the chloroplast. Remove the chloroplast. The second water bath is chloroplast removed. The chloroplast is removed. Chloroplast removed. It's a hydrated gel. Cover the gel. Careless. Careless. After this, produce two hormone. Produce two hormone. First one is auxin. Second one is cytokinin. The auxin is a low pressure hormone. Cytokinin high pressure. Now use the cytokinin for high pressure cytokinin. Cytokinin. It's a high pressure. Mitosis, division. To produce new plant. To produce new plant. To produce new plant is it's a new plant. After this, seed become ready. Now ending the lecture number 5, tissue culture. It's a tissue culture structure. Now see the tissue culture structure. End of this lecture.